Okay, so in the last video we were talking about the structure and the mechanism of the action potential, and we kind of introduced you to the idea of the synaptic connection. So the connection from the synaptic bouton of one neuron where the action potential must end, and the dendrite of another neuron. So the way that two neurons can speak to each other is through this chemical connection called the synapse. So let's have a look in a bit more detail at the synapse. So here we can see the diagram we had last time. So here is our synapse. Sometimes you will see it referred to as a synaptic junction. So I will label it thus this time. And this little gap you can see here, there's a gap between a certain distance. This is called the synaptic cleft. There it is. So that's the gap between the bouton and the dendrite of the neuron, the next neuron. So let's spend a bit of time looking at the synapse in more detail. So this is a very simplified diagram of a synapse, and there's a number of parts that we need to know about and need to understand. So here we can see the synaptic cleft. So you've got the gap between them here. There's your cleft. Now this neuron here, so this is the synaptic bouton of, this is the bouton, you know it, of coming from the axon, so the action potential is traveling in this direction. Now this neuron, in this case, we call this the pre-synaptic neuron, because the information is traveling in this direction, and this is, as you would expect, this one here is called the post-synaptic neuron. Now there are a few other components here that we need to talk about. So you will notice here I've got these little circles. Now these are known as, where do I draw this? I'm going to do this in red, why not? So these are called synaptic vesicles. Now a vesicle generally is simply refers to a bubble, a spherical bubble of membrane that are normally found inside cells, and these very small bubbles of membrane that you find in the synaptic bouton are a specialized type of vesicle known as the synaptic vesicle, and they're specialized because they contain, or they are filled with, special types of molecules. A small number of which we will be looking at in more detail, and these molecules are known as neurotransmitters. So this yellow circle here, which I have now reddened, is a neurotransmitter. You might well, of course, heard of this term, the idea of a neurotransmitter. And the function of the neurotransmitter, as we will see, is revealed by the name. It transmits information from the presynaptic to the postsynaptic neuron. Now, in this highly simplified diagram, you've got two vesicles here, and each vesicle contains one molecule of neurotransmitter here. Now, in reality, each vesicle, and there might be up to kind of 300 of these synaptic vesicles in a single bouton, each vesicle may contain up to sort of 10,000 of these neurotransmitter molecules, but in this simplified diagram I've shown only one to keep things hopefully completely clear. So what's happening here? So let's maybe number this. So here we have number 1. This is where the neurotransmitter is found outside of the neuron. And neurotransmitters are often or sometimes made outside the neuron, sometimes inside the neuron, but they need to be brought into the neuron here through these special channels and then packed. So here's our neurotransmitter now inside the neuron, and they need to be packed into the synaptic vesicles. The synaptic vesicles need to be filled with these neurotransmitters. So that's what's going on here. So label this 2 and 3. So you have an empty vesicle, number 2 here, and then it's filled up with neurotransmitter in 3. Now, what are these vesicles for and what are the neurotransmitters for? Well, when an action potential actually reaches the bouton, it can go no further. There's no way, as I said before, for it to jump across the synaptic cleft. So what actually happens is the action potential causes these vesicles to kind of stick to the membrane. Now, there's actually normally a lot of these vesicles already kind of very close to the membrane, like this. And they cause the vesicle to actually fuse with the membrane of the synaptic bouton. It's useful actually to maybe draw this because it's kind of interesting. So this is the membrane and this is a vesicle. When a vesicle approaches the membrane, it actually sticks to the membrane. It's called docking. And then here, the two membranes actually fuse together. So it goes kind of like this. Then it fully opens out like this. So if we draw in now the neurotransmitter in yellow, so the neurotransmitter is in this bubble of membrane, in this synaptic vesicle. It's inside the cell, and now it is outside the cell, so it can escape. So now we can see what's going on here. So here we see the neurotransmitter has been released from the presynaptic terminal, as it's also called, the bouton, and will then diffuse across the synaptic cleft and bind to special receptors that are found on the postsynaptic cell. So here in green I've drawn these receptors. So this is a receptor. Now receptors we will talk about in a lot of detail later on because receptors are really important for understanding psychedelic drugs. So the receptors lie on what's called the postsynaptic membrane here, postsynaptic membrane. And when the neurotransmitter binds to a receptor, so here you can see a neurotransmitter has bound to a receptor, and it looks like here it's just about to bind to the receptor. It has some kind of effect in the postsynaptic neuron. In other words, the postsynaptic neuron receives information in the form of this chemical called the neurotransmitter. So we go from, if we look at this more globally, we go from a, let's maybe draw this. So this is electrochemical, i.e. the action potential. Then this is all chemical. So it's just the diffusion, the release and diffusion of neurotransmitter. And then maybe, you will get another action potential in this, the postsynaptic neurons. It becomes again, electrochemical. So neurons neurons generate electrochemical information in the form of the action potential that is transmitted chemically across the synapse using these neurotransmitters. So this then raises the question is what do these neurotransmitters do after they are released and after they release from the presynaptic neuron from these synaptic vesicles, what effect do they actually have on the postsynaptic neuron? And that will depend upon the type of neuron and the type of neurotransmitter that it releases. So there are 100 or more neurotransmitters that have been identified. We're going to look at a very small number. In fact, 2 key neurotransmitters we will look at, as well as some related molecules known as neuromodulators, which are similar idea but work slightly differently. But generally, one type of neuron will generally release, in other words, the neuron's axon, the synaptic bouton, will generally release a single type of neurotransmitter. So let's have a look at the types of neurotransmitters and then think about how they actually work. What is the effect of these neurotransmitters on the postsynaptic neuron? Okay, so in this diagram from compoundchem.com, very useful, very pretty diagram. Now, don't worry so much about the labels here they've given them. Now I'm going to cross some of these out, not because they're not important, but because we're not going to discuss them. But first of all, just go through them. So adrenaline, you may well have heard of here. So we've got adrenaline here. Noradrenaline, maybe you've heard of that as well. Dopamine, serotonin, you almost certainly will have heard of that. This one called GABA. It's not a type of dance music. Well, it is a type of dance music, but it's also a neurotransmitter. Acetylcholine, glutamate, and endorphins. Now, these are all neurotransmitters in that, what defines a neurotransmitter? Well, we've kind of defined a neurotransmitter. A neurotransmitter is a chemical that's released from the synaptic bouton, that's stored inside synaptic vesicles and then is released into the synaptic cleft from the bouton in response to an action potential. Okay, so which of these neurotransmitters are we going to look at? Well, we're going to look mainly going to focus on this one here called glutamate. This is a really extremely important neurotransmitter. We'll also look at GABA, and later on we will talk about serotonin. That's really important when it comes to the actions of psychedelic drugs. But for this video, we're going to talk only about glutamate and GABA. Let's start by talking about glutamate. So glutamate is a neurotransmitter that is released from neurons, certain types of neurons that are called glutamatergic neurons. So these are neurons that release glutamate, as you'd expect. And glutamate binds to particular receptors. Remember these postsynaptic receptors in the postsynaptic membrane on the opposite side of the synaptic cleft. Glutamate, when it's released from the presynaptic bouton, diffuses across and binds to specific types of glutamate receptors. Now, to understand the effect that this has on the postsynaptic neuron, we need to look at one particular type of glutamate receptor. We'll also talk about GABA as well. So let's have a look here. So this diagram from Nature shows 3 types of receptor. Now, we're going to ignore this receptor here, forget about him, and forget about him for a while. We're going to talk about this receptor here, and this is called, for reasons that we won't go into, the AMPA receptor, or the AMPA receptor. And you can see on this diagram that this appears to be some kind of channel. So you can see it has this cylindrical structure, and there seems to be a hole here, yes, through which sodium ions are traveling, and glutamate is indeed bound. So let's have a look at the glutamate receptor in a little bit more detail. Okay, so again, let's draw a line for our membrane. So receptors sit in the membrane, and we'll talk a lot about receptors, I think, in Unit 3. But briefly at this point, all receptors are proteins, of course, and they all have basically 3 regions. Again, I'm drawing it like this in 2 parts to show that it's a receptor, but it's also a channel. So there are basically 3 regions. So this region here, I'm going to colour yellow, is referred to as the extracellular domain, because it sits outside the cell, and it's the extracellular domain where glutamate binds. So let's draw in glutamate here in orange. This is glutamate. So I will always represent just as glue. Then you have another region, which I'm going to do in green here. This is referred to as the transmembrane domain, because it traverses the membrane. It goes through the membrane. And then, as you'd expect, or as you can perhaps anticipate, what are we going for here? I don't like brown. Let's go with light blue. Here, this is the intracellular domain. And so overall, the receptor spans the entire membrane from outside to in. And this is why these channels, so this is a receptor and a channel, remember, allows things sometimes to move through. Now, not all receptors are channels, and we're going to talk about a very specific type of receptor later on in the course, the serotonin receptor or serotonin receptors, which are not channels, but this is a channel. And it's a special type of channel. So already we've met the voltage-gated channels, right? We met the voltage-gated sodium channel, which allows sodium to pass through when it's open, and it's opened by voltage, by the membrane potential reaching this threshold, and the voltage-gated potassium channels, of course. But this is a different type of channel, the glutamate receptor here, well, this type of glutamate receptor, I should say, there are a number of types, is called a ligand-gated ion channel. A ligand is simply a molecule that binds to a protein, basically. So in this case, the ligand is the glutamate molecule. So how does it function? Well, it should be pretty intuitive what's going on. When glutamate binds to the channel, and I think I will get rid of this, like this. So when glutamate binds, it causes the channel to open. So this is an open form of the channel. So I'm not doing the color coding here. I will put glutamate in there so that's clear. What am I coloring? made in blue anyway. So I should, of course, not. So this is closed. So there's nothing flowing here. However, here we can see on the right, the channel has opened. Now, what does that mean? It means that ions can now flow through the channel. Which type of ions? We've met them before. Sodium ions. So the glutamate receptor, or this particular type of glutamate receptor, and this is the AMPA receptor, is a ligand-gated ion channel that is gated by glutamate. So, let's think about this. So glutamate is released from the presynaptic neuron, diffuses across the synaptic cleft. It binds to this special type of glutamate receptor called the AMPA receptor, which causes the AMPA receptor, which is this ligand-gated ion channel, to open and allows sodium ions to flow through the channel. Now, in which direction are sodium channels going to flow? Are they going to flow out of the cell? Or they're going to flow into the cell? They're going to flow into the cell, of course they are. Because we know already from the first video that there is a high concentration of sodium on the outside of the cell. So sodium ions will flow in whilst these channels are briefly open. They're briefly open because glutamate will bind, and then it will unbind quite rapidly, and the channel will close again. But in that brief period of time when the AMPA receptor channel pore, the little hole that goes through the channel, is open, sodium ions will flow into the cell.